All right, everyone, welcome to this week's biology investigation. This is the week of February the 21st. I'm Shannon Reeves. That's Ravioli. She's being a perfect little loaf for us all. And you are either in section 39 or section 40. So let's go ahead and get started with some biology. First, we have some announcements. The 5-minute meetings. We know that there are like no slots available. That is some weird setting on Calendly, so I'm doing my best to try and wiggle around it. And I've maybe met with like a fourth of you now. So there's a lot. And it's going to take a few weeks for me to get through. So don't worry if you haven't signed up for one yet. There will be more to come. The deadline won't happen until everybody's met with me or has had an opportunity to meet with me. So, the other thing I would like to say is though, be respectful of the times that are available. Try not to sign up for more than one, or try not to schedule one and then not show up. So, that's something that's been happening quite a bit. And if it keeps happening, then we'll have to say that you get one opportunity to meet with me. And if you don't show up, then you don't show up. So just the more respectful you are, the less likely it is that we have to set up rules like that. So I've been getting a lot of frequently asked questions this week from students, and I'd like to bring some of them up in the PowerPoint so that maybe we can address them as a whole. So you're writing summaries now, and you're a little bit more familiar with what's expected from you to complete your assignments. So one of the things that you'll be using is this lab manual and course resource tab. You've probably used it a little bit on this last one, but you will use it a whole lot more this semester. So start looking at that top tab. This should be what you guys view on Canvas. But go ahead and utilize that top tab a little bit more, and there will be a lot of resources about how to write your summary. Namely, two that I keep pointing out this week are, this is really, really small, but this is how to properly cite, and this one is how to write a lab summary. These are two things that you can use the whole semester long that will help you out and will save me a little bit of time with emails. So I would say before you write me an email, go ahead and check this tab to see if there already is a resource there for you that might answer your question. But of course, if you still can't find it, feel free to reach out to me. So the other thing is make sure to check your modules entirely. This week there's 4 steps to completing this assignment. So this is just a little reminder that I expect you to do a little bit of discovery on your own and navigating Canvas and figuring out where things are before emailing me. So now from here on out, I'll probably write some sort of an email in a response of, have you checked the modules on Canvas, rather than giving you an explanation of where things are. All right, so students are so worried about this word count, and I do not blame you. 250 to 300 words is such a small amount. But I want to remind you all that you are writing lab summaries, not lab reports. A lot of students have already had chemistry, or maybe some of you are juniors or seniors, and you've already had several sciences, so you're well acquainted with the lab report format, and that is not what we're writing. We're writing more of an abstract that is like a summary that covers the entire lab report. So 250 words to 300 is actually not really unreasonable. But that means really stick to the script. All of those points that we have in the rubric, make sure and really cut the fat from everything that you have to say. So often whenever you submit scientific papers for publication, you're required to limit the summary to your experiment. And part of scientific writing is learning the art of brevity, being able to be concise and direct. It's not like an English essay where you have a lot of flowery language and a lot of use of metaphor, or a lot of I'm kind of getting onto the flowery side of writing detailed essays. This is very direct and straightforward. So just keep in mind, I'm actually a little bit lenient on that word count. If you go over but you've hit all of your marks on the rubric and you feel like you've been doing a good job of meeting all of that, then I'm going to I'm going to go ahead and say you're fine. But if your lab report is 900 words, but it doesn't necessarily hit all of the points in the rubric, then that's too much for not addressing all of the points. But on the other side of the spectrum, if you're within 300 words, but you don't hit any of the parts in the rubric, then that will be points off as well. So really, I'm more driven by content that you have than I am word count, but know that respecting the word count is something that we want you guys to try and learn to do. So, moving on. You are in section 39 or section 40. So we figured out, thank you to Isabel, we figured out that your Canvas page said forever that you were in 37, section 37, simply because Canvas makes us put a placeholder there, but we didn't think this was visible. So we changed it to Online Lab. It should say that. If it does not say it, you might not be in my section, but email me just in case. By now, I think everybody's figured out what section they are and that I'm your TA, but there's always confusion. So feel free to email to figure that stuff out. But as another point, where are your office hours and where's the link to it? I simply don't send that out because of privacy. So it has nothing to do with not wanting to be in office hours or meet with you guys. It's because there are some students that want to meet one-on-one and feel comfortable asking those questions on one-on-one. And if I give an open door, then typically those students don't speak out and get the help that they need. So I am just respecting that. And so if you want to meet with me, by all means, even if it's last minute and you say, hey, I'd like to meet in your office hours right now, I'll send you that link. Just make sure and email me either before or during so I can send it to you. All right, so let's talk about this week. What will you need to do before leaving your computer today? So we're going to carry out experimental procedures, collect and display data. You're going to determine how enzyme activity is affected by several different conditions, and you will write a lab summary, upload it to Canvas. This doesn't say it, but you also need to do a Flipgrid. All right, so helpful tips for today's investigation. Why do some fruits and vegetables turn brown faster than others? So this is a question that we will pose to you guys as students, but this necessarily isn't the research question that we want you guys to formulate. We'll talk a little bit more about it, but this is essentially the question of the investigation. So even though you're not in lab today, you will have, you'll probably want to know about the equipment that we use and the procedures that we go through just so that you can write about them in your methods. We are going to be using a colorimeter. You should have watched in the background that there is a colorimeter video that tells you a little bit about the equipment that we're using. We'll be using 5 cuvettes. They're little tiny see-through containers that go inside of the colorimeter, and then tubes to contain our enzyme potato slushy, and 4 droppers to add the reagents. So typically in lab, what happens is that I will be preparing this potato solution in the background. It's usually just like chopped up potato and deionized water. But as a side note, why would it be important to add deionized water? You might think back to lecture, what are ions and what property do ions have that would be crucial to either include or exclude them from certain chemical experiments. So I will be making this potato slushy, and essentially this potato is just potato and some water, and it will over time turn brown because that's what potatoes do, right? So some of you may have had, everybody's opened an apple and see it get brown over time, right? A potato. You may have had a mom or a dad that for your school lunches put squeezed lemon juice on top of it, or put apples in the refrigerator after they were sliced. So there's a reason why you go to Walmart and Target and they have those sliced apples, or I think even McDonald's offers sliced apples in their Happy Meals now, but they aren't brown yet. And that is an interesting, there's an interesting reason behind that. And that's actually a big market today that people want to figure out. How do we prevent these things so that we can sell food and get it to people faster before it goes brown? So you'll be doing an experiment with potato solution to see how fast it turns brown, essentially. You are going to be comparing 3 different environments, though. So you will have salt, a salt environment where we give you, I think, 3 or 4 different salinity levels, different concentrations of salt. You will be given pH, where you will have an acid, a neutral, and a base. Um, Sometimes we give you 2 different levels of acids or two different levels of bases. And then the next one is temperature, where you will be using hot, cold, and then room temperature baths. So essentially, I'm going to go back here. Actually, oh shoot, I can't go back. I don't know how to do this, and I'm deep into this video, so I'm not going to stop it. I'm just going to keep talking, so ignore this slide for a second. But we have these 3 environments. One is salt, one is pH, one is temperature. So essentially, you're actually kind of doing 3 experiments this week. So you will want to create a hypothesis for each one of those scenarios. Again, word count. Yikes. It adds up here whenever we have kind of 3 experiments in one. So again, don't panic about the word count if your content is good. But you will want to have 3 different hypotheses, very similar to the macromolecule lab. 1 for salt, what do you think is going to happen? 1 for pH, and 1 for temperature. This is just a reminder on this slide about being safe in the lab, using proper equipment so that you don't hurt yourself, essentially. This is something that seems silly to include in slides for an online lab, but you would be surprised how quickly data collection is ruined because students don't realize the importance of labeling all of your data. So this is actually a good slide to remember. Even though you're not doing this, label your data in your online lab stuff too. Often students will write down a list of maybe results or values for a group and totally mix it up with the other group because they simply didn't label it on their piece of paper. And then their graph looks weird and then they lose points. So just a reminder to be really thorough about how descriptive you are labeling different things like that. So what happens is that after 10 minutes of adding solution, so you will have this enzyme in tubes, the potato solution in tubes, you add your different environments. So you will either add some pH, like add some acid, add a base, add a neutral, or you will add some salt concentrations, different salt concentrations, or you will put them inside of the baths, the hot water bath, the cool ice water bath. And then after 10 minutes, we add this catechol to our tubes. So our samples already contain the enzyme catecholase. This is something that helps us understand how much the enzyme is working. So we will be adding that, and then after 1 minute, you're going to put, here's the little cuvette on the picture on the right. I don't know if you can actually see my mouse, but the picture on the right shows the cuvette, and we put the solution in there. It's actually really little, so you just put a little bit in there, maybe like 1 milliliter or 2, inside of the cuvette, and then put that inside of the colorimeter. So the colorimeter is a pretty interesting instrument. Here's a picture of someone putting the drops from the tube in there. But notice that the solution in the right picture is somewhat cloudy, but it's not really brown. So we want to make sure and impress upon you the relationship with what we're looking at, which is our transmittance and the enzyme reaction. So if your enzyme is going to be brown, then it's working. Or I'm sorry, if your potato solution is brown, your enzyme is working. That means the enzyme's whole job is to turn the potato brown. If it turns brown, it's good. But if it's white or see-through like this, then your enzyme rate has slowed down. So this particular cuvette, though, on the right is particularly see-through. That is the amount of transmittance. So you would expect its transmittance to be rather high because light can shine through it. So keep in mind that it's kind of an opposite situation, that whenever you have high transmittance, it means your enzyme's not really working because it didn't turn brown. And whenever you have low transmittance, it means your enzyme is working because it has turned brown. So just be careful with that interpretation in your lab summary at the end. So you can record the transmittance. There's a video, the online simulation of Dr. Shaw and I doing this lab for you guys. And we put the transmittance on the computer so that you could see. It will wiggle around to numbers, and it may even change whole numbers or just decimals. But take your best guess at where it stands at. You don't have to be super exact with this. And you can round up to 1 or 2 decimals. It's kind of up to you. But just keep that in mind, that you don't have to have one number, and not everybody has to have the same number. So after we're done, we just want to remind that we are all super helpers, and we will clean up after we're done, and we don't leave messes. A reminder that you can clean out droppers by just squeezing soapy water in and out of it. You don't have to hold it underneath a faucet and try and get water inside. I've seen that happen before in my lab, and it is a waste of time and a waste of water. So finally, we'll go ahead and rinse everything when we're done and leave everything organized for the next class. Okay, so let's move on to writing hypotheses. So remember that your hypothesis is your causal explanation. So you want to make sure, she's so cute. Look, she's just like a little pumpkin. Okay. A hypothesis needs a causal explanation. So keep that in mind, that you need to have that. But it just has to be testable. It doesn't have to be correct. If you don't know what salt's going to do to a potato, that's okay. Make an educated guess. What do you think salt does in general? We've been talking a lot about salinity in lecture. So what is the role of salt? What is its relationship with water? What do you think that there are some properties about salt that we might have talked about with redox reactions that may play a part in enzyme reactions? Keep in mind an enzyme's a protein, so proteins are pretty fragile to denaturation. So we have an example hypothesis right here. Enzymes exposed to pH of 11 have lower activity because bases denature enzymes. You don't have to support it, you just have to write about it. And that's what you're graded on, is how you write about it. So you do need 3 hypotheses that you'll include in your lab summary, which also means you'll need 3 graphs. Just keep that in mind, just like the macromolecule. So reminder, no more tables. I think I have a slide in just a second that talks about that. But you will have 3 different graphs, just because we're doing three or four conditions in each of those. In the macromolecules, we did have 4 hypotheses, but we were doing repeatable conditions each time. So that's why we only had 1 table. But I wanted to go over some parts of the lab report that may help you. I know that sometimes the wording in the rubric can be a little bit confusing, or if you have had another science, they may have different requirements for what their introductions, methods, results, and conclusions are required. So I figured Ravioli is going to teach us, because of course. Also, look how blurry that picture is. Did I take it in 1992? Oh boy, that's so blurry. Okay, so let's say we had an experiment with ravioli. We're going to go over the little parts of the introduction with this, but just as a reminder, her favorite thing is food, number 1, not me, I wish. Her second favorite thing is food, and she likes food definitely more than me. And then her third thing is sleep. So food, sleep, that's her favorite. But what we want to know is ravioli sleeps in specific spots. We've noticed this, me and my husband. So if ravioli likes to sleep in different spots, then what draws her to these spots? We have like a hypothesis, or the beginnings, maybe a research question. We think it might have to do with the temperature of the location, because we know that it's a pretty big historical fact/myth, anecdote, I don't know, that kings and queens used to keep cats around so that they could chase mice and find the hottest seat in the castle. So we thought, so right over here on the left, you can see, oh, she's so smush. She's in her little kitty cat bed, and she likes to sleep right there. She also likes to sleep on our bed where the light shines, and then she likes to sleep in a box too, even though we bought her that bed. She prefers a free Amazon box. But one thing that we did notice is this is her right here being so cute. There's a vent right there. So we think, I think we're on the right track. So the introduction wants you to, the first part of it, address the study's major question. Why was the work done? This can be done in like 1 or 2 sentences, really. So we put, Ravioli enjoys sleeping almost as much as eating. It is one of her favorite hobbies. We've noticed that there are specific spots around our house that she favors. In the past, sunshine and warmth have been a common factor in the spots that Ravioli picks. That's enough background, right? So let's move on. Oh my goodness, it's so adorable, her and her brother. She's so generous with how she's willing to share her sleeping spots with her brother, even though her brother is like 4 times the size of her. So we want to create our own question rather than the generic question at the beginning of the slides, like I talked about, because that's just not specific enough. So your question may have something to do with comparing environments, such as, does pH level have more of an effect on enzyme reaction than temperature does? I don't know, let's find out. Or you may want to focus on a question like, do hot temperatures have the same effect on enzyme reaction as cold temperatures? That could be something that you want to talk about too. But you'll want to make sure and answer that question with your hypothesis. So what do you really want to know from the experiment? Don't use general questions that we give you and answer your research question with the hypothesis. So we put, we're curious if warmer temperatures are what attract Ravioli to her favorite snuggle spots. We hypothesize Ravioli's sleep spots have higher temperatures throughout the day than other areas of the home. Again, 2 sentences, short and sweet. So far, I think I had 3 sentences in the previous slide. We're up to 5. And again, that's wordy. So okay, so how the work was carried out. This is the last thing in the introduction that we want you to focus on. So address the design and the experiment in broad strokes. This doesn't mean give me all the details. That's for methods. And I know that's a confusing thing because we want to talk about how the work was carried out, but it's more like the procedures, like how much of each liquid did you use, or how long did you test each sample for, all the minutia like that. So in our experiment, we're going to give a general statement that says this experiment was carried out by taking the air temperature in the areas Ravioli was taking naps and comparing those temperatures to other spots in the house. Ravioli was not pleased or cooperative during the process, hence the grumpy pictures. All right, so then we have an introduction complete. I'm not going to read the whole thing because we've already read it, but those are the 3 points essentially that we want you to do in your hypothesis. So what are you addressing? What's the background that we need to know? That's our first, just a couple of sentences. 2, research question and hypothesis, 2 sentences right there. And then last one, how did you carry it out in general, not specifically? So we can have a pretty concise introduction. Now, if you were writing a lab report, this would be 5 paragraphs. It would be a fleshed out version of all of this with citations also. But that's not what we're doing in this lab. We're just requiring lab summaries. So speaking of citations, we're going to have you do some more citation stuff. But keep in mind, here are some tips for paraphrasing scholarly literature. So is paraphrasing the same as quoting? No, don't quote. That is English. Again, there's different formats for different studies in academia and in science. You don't quote, you paraphrase. So summarize into 1 sentence. That's another important thing, is that if you use a citation, you don't use 5 sentences from the citation. You can make a sentence that's concise about those 5 sentences that is in your words, and that's paraphrasing. So then you put the in-text citation, the name of the article, and the year afterwards in parentheses. So we're actually going to have you do that for this one, this lab summary. So right here, paraphrasing practice, we have an article about enzyme browning, and we'll give you the citation information in the next slide. But essentially, we want you to take this paragraph, paraphrase it into 1 sentence, not a run-on sentence, not a sentence with a semicolon, 1 sentence that is pretty concise, and then cite it. You will have to have a literature cited portion in your lab summary again. So this will be on Canvas so that you don't have to come back to this part in the video and actually pause it and read it and paraphrase it. So we'll have that on Canvas for you so that you can access that easily. And here is your information for the article. So keep in mind that how to properly cite article in Canvas, that PDF that we have for you, will tell you exactly what you need to do for this citation. And in case anybody has questions, it's APA formatting. All right, so this was the reminder that I was talking about. No tables this week, return to making graphs. You will never do a table ever again. If you have a table in your lab summary, it will be a 0 for that portion of your lab summary. We need to have graphs, and we need to know how to make good graphs. So this is my graph for the ravioli scenario. So one hypothesis per condition, which means you're making 3 graphs. So notice that I put my data, my dependent variable here, and then this is my independent variable with my different groups, and you will just have one type of data here. I think you only get one translucent or transmittance data point. So keep in mind that you can't make a box and whisker plot from that. You need at least 4 pieces of data to make a box and whisker plot. So you'll probably be making a bar graph, this lab summary for each of them. And this is what my bar graph might look like. So whenever I put percents inside of the cells, they'll come up as percents automatically on the graph, and that's okay. So you can do that. I've added data labels. If you right click on your graph, you'll see format data labels, and you can add them or add data labels. Notice that I have a title that's not Enzyme Lab. I have my independent variable, which is my x-axis title, and y-axis title is over here. So keep in mind that the axis title is not the same as group 1, 2, and 3. I didn't add a legend simply because I didn't have any actual data here, but you would probably have a legend over here or underneath or wherever. But just keep in mind that you need to have both identification of your groups right here or somewhere and your x-axis title. But since they're both usually on the bottom, sometimes students forget those. So just a little review. This is our last slide. R6 is how to cite. R6 probably doesn't make a lot of sense to you, but class resources and lab manual, the R means resources. So there is a PDF called R6 how to cite and the how to write a lab summary document that are in Canvas there to help you. And that your summary is due at 5 p.m. on Friday, like all of the other ones. Keep in mind that I want you to do a Flipgrid video. You'll continue to do Flipgrid videos from here on out and to report what you think. So keep in mind that I'll have a lot of that information on Flipgrid about what I want you to say for this week's Enzyme Lab. So if you have any questions, reach out. I'm here. Try and check Canvas first to see if you can discover anything on your own, but then if you can't, I'll help you out. And if you want to sign up for office hour times or the 5-minute meetings, keep an eye out that I'm going to send an email out with other times that I do have available for this week coming up on Tuesday and Friday. So stay safe and warm out there, and I'll see you guys next week.